So, the next talk is by Ranjith Padinhateeri from IIT Bombay. Excuse my pronunciation of the name. My name is no easier to pronounce, actually. So, Ranjith is going to talk to us about the nature of intra-chromatin interactions in the three-dimensional folding of chromatin.
Can you hear me at the back? Yeah, thank you. And thank you for the organizers, Devay and Sandeep, for inviting me. So I'm going to talk about three-dimensional folding of chromatin, and some of you, so I was just telling that I have given this talk in Max Science before, so all the Max Science people might have heard at least first part of the talk, but there is something at the end which is new, which is a little bit newer than what I gave last time. So let me just quickly go through what three-dimensional folding of chromatin is. So the word condensate or condensation has been used in the context of chromatin for the last 40 years or so. So chromatin condensation is a known phenomenon where the DNA, which is the bare DNA, condenses into a structure which we call chromosomes or chromatin, right? So DNA is not bare in the cell, it is condensed by large number of proteins into forms of chromatin as shown in this electron microscopy picture here and kind of a Gen AI picture there. Now, the question is, what is the nature of condensation? And it turns out that whatever the specific nature or the way the DNA condenses decides the fate of the cell. For example, if the cell becomes a neuronal cell or the cell becomes a skin cell is decided by the way the DNA locally condenses. So therefore it is important to understand how it is condenses in space and time. So it has a very specific spatial organization the way it condenses, and it has also a temporal organization, and all of this matters for all the processes like gene regulation, recombination, translocation, repair, spreading chromosome modification, and so many different processes. The 3D organization and the local condensation matters. And one of the experimental method to understand the precise nature of how it condenses or locally folds is a method called chromatin conformation capture experiments. And let me quickly tell you what this is. So basically, DNA is some polymer, and as it folds, different parts of the polymers will come together and interact, and this method allows you to figure out these contact points. So what you have is that you add a cross-linking agent that cross-links wherever they are folded and in contact, and then you can add a enzyme that cut the DNA at random locations, and that would leave and separate, that would make a pieces of DNA like X. For example, it cuts here, 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 and here. It would generate a piece like X, which is a piece of DNA, where this is one part of the DNA and this is other part of the DNA. And then you can separate it out, and then you can sequence. Then you can sequence that X-like part. So this part might come from one region of the DNA, and while the other part might come from far away region of the genome. And these two X and Y can be in contact, and these experiments can measure what is the probability that X and Y was in contact, and for any X and any Y, that is like a matrix. So you can see that experiments can generate a contact matrix which says that what is the probability that a segment X was in contact with segment Y. So this is the Y and this is X, right? So this is for chromosome X, some particular region 90 megabase to 100 some megabase, both same, and this is the contact matrix, and this squares here says that they are locally condensed. So you can see that they are locally condensed here, 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 and these two regions again interact, forming this kind of interactions. So there is, this already tells you that there is some condensation plus some few condensates interact together. That's what this broadly tells you. So once experiments can generate this contact structure. Now the question is, given this kind of a contact map, how do you go back and generate a 3D structure? Because experiments can only provide a two-dimensional structure, contact map like this, and from this you have to generate a three-dimensional structure. And it needs some polymer physics, and we have been thinking about how to go from here to here, and this is something that we have been working for last few years. One of the important points to remember while going from here to here is that the contact probability between segments I and segment J is always very small, typically like less than 10%. So this means that it is highly dynamic structure. Things are like in some senses liquid-like if you wish, and things are changing with time, and therefore it is not trivial to go from this picture to that picture. Yeah.
You measure the NOEs and then you use the strength of the coupling to determine distances.
Yeah.
And then that's on a two-dimensional map.
Yeah.
And then you use those as distance constraints in modeling the three-dimensional structure.
Yeah.
So why is this going to be so different?
So it is different for a few reasons. First of all, this is an active problem where there is enzymes doing stuff all the time. And the interactions are much more complex. Like there's not like 20 amino acids. Here there's like so many different types of interactions, and the interacting partners itself are binding and dissociating. Their concentrations are changing in time, and they are all active. So there is, if you look at the entire picture, is much more complex. But I fully agree that the idea of protein and how the protein fold can be very useful to think about it, and we will use some of those ideas to think about it as well. Like you will see that some of those ideas that we used was also used for protein folding. So it is useful, but if you want to solve the full problem, this is much more complex. So yeah, so you need a polymer description of chromatin, and we understand the polymer description of DNA reasonably well. DNA has a worm-like chain model, and we know the persistence length and so on and so forth, but we don't know much about the polymer property of the chromatin at all. So, and if you want to simulate chromatin, if you want to start somewhere, you have to start with the polymer. And what would you put the polymer property? If you want to generate, if you want to simulate it on a computer, what will you put as the polymer parameters? And actually, there is people heuristically simulate chromatin, but there is no real idea on what those numbers, actual relevant numbers are, and something that we try to work out in detail. So, just to quickly take you through this, chromatin is a complex polymer. There is first there is a DNA which is folded with the help of a protein called histone to form this kind of a structure, and then those histones further interact and form a higher order structure, right? So now there are many types of interaction. As you can see, there are different colors of proteins, and we don't know what their interaction strengths are, how strongly they interact. All of this information we have no idea at all. And now if you want to, not only that, if you want to model chromatin, you cannot model at the nucleosome resolution. So the natural way of modeling chromatin is, you can say that, okay, this is my polymer and nucleosome is like a bead, and if I do that, one human chromosome is like 1 million nucleosomes. So you cannot simulate 1 million beads. So you cannot even simulate a single chromosome, and we have 46 chromosomes. So that's like 46 million of the order of that kind of beads, and it is impossible to simulate the whole genome as at the scale of nucleosomes. So one has to go into a much more coarse-grained scale where multiple nucleosomes are like considered as a single bead. So any realistic simulation would typically require a bead spring polymer chain where one bead represents a chunk of chromatin, and that chunk could be 1 kb if you want to study a gene region, and that chunk could be like 100 kb or 1 megabase if you want to study the whole genome. So depending on what problem you want to solve, you would use a coarse grain model for chromatin, and the coarse graining level will depend on what exactly the question that you want to answer. But how do you coarse grain this kind of a heterogeneous polymer? That's a question. And typically, let's say models assume a few kb is equal to 1 bead. So, but if you assume, for example, this is 6 nucleosomes. So if you assume 6 nucleosomes as 1 bead, we don't know what is the size of this bead. Is it like 10 nanometer? Is it like 20 nanometer? Depending on how these 6 nucleosomes are folded. If the 6 nucleosomes are like this, then the bead will have a different size. If those 6 nucleosomes are folded like this bead will have a different size. So even the physical dimension of the bead is not easy, and it's not easy to understand. And this is a question that what is the physical dimension of the bead? Yeah. So we want to systematically coarse grain then. So if you want to coarse grain, and once we coarse grain, then if you have a coarse grain model, in the coarse grain model we want to know what is the intra-chromatin interaction. For example, one region of the chromatin would interact with other region. What is the strength of this interaction? Not only the strength, what is the nature of the interaction? What potential will you put in there? Would you put a Lennard-Jones potential, or would you put some other potential? And what is the strength? What are the parameters of those potentials? So there are two questions about the intra-chromatin interaction. And not only the intra-chromatin interaction, what would you put the spring constant as? Like, is the spring very flexible, or is it kind of a rigid, stiff spring? We don't even know that because we don't know. Nobody has pulled chromatin at the scale of a gene. All those people who have pulled chromatin have taken the whole chromatin and pulled at the scale of a mitotic chromosome or something, but nobody has pulled at the scale of a gene by taking an in vivo chromatin. It's not at all very easy to do it. Okay, so, and there are many other parameters. For example, the bendability of chromatin. Is chromatin a semi-flexible polymer, or is it a kind of a more flexible polymer? If it is a semi-flexible polymer, what is the bending stiffness of the chromatin? Does it have an intrinsic curvature? So none of those parameters related to chromatin is not known. And something that I want to quickly tell you today is how we determine, or we have some estimate of these parameters, and in particular I want to focus on the nature of intrachromatin interaction because that drives a lot of condensate formation and so on and so forth. Okay, now what we know, so I said a lot of things we don't know. What we know the last 30 years of literature is something at the nucleosome resolution. Thanks to many crystal structures and so on and so forth, we know that nucleosome has a dimension of about 10-11 nanometer, and we know that how often the nucleosomes are placed on an average. So we know the typical nucleosome distance and so on and so forth. So since we know if we have a chromatin model at the resolution of a nucleosome, we know a lot of length scales. So what we did was that we started with a contact map data that gave us contact map at the nucleosome resolution. So there is a very specific experimental data called micro-C experimental data, which uses micrococcal nuclease. That's why it's the name micro-C, and it generates contact map at the resolution of a nucleosome. So this is, for example, the data from C et al. Here for example, this dot represents a nucleosome at 31.22 megabase is in contact with another nucleosome at 31.24 megabase. That's what this means. So every dot in this is a contact between two nucleosomes on an average typically. And what we did, so Sangram, who was in my lab, as a PhD student, we made a model to go from this kind of a picture to a polymer picture. Actually, we have two models, and I wouldn't describe the details of this model now. I will be happy to discuss it after the talk because for the wanting of time. But one of the model is the following, that we generate an ensemble of static chromatin conformation, and the average of those ensemble of conformations should give you this contact map. Now we have a particular procedure and it has some details, but the crux of that is that you can put strongly interactions like springs, which would bring together faraway beads. For example, this dot would mean I would put a spring between this and bring them together. But the spring is inserted with a probability Pij. It's not that always the spring is there. The spring is there or not there stochastically with the probability Pij. So this brings in the stochasticity into this, and the Pij putting in would essentially at the end of it generate this kind of a contact map. So we go around this, follow some procedure, which is a little bit non-trivial procedure, and then generate a large number of static conformations and then equilibrate those conformations, and the average of those large number of ensemble of conformation would give rise to this kind of a contact map. For example, this is the experimental contact map, and this, if you do the procedure, you will get a similar contact map. For example, there is a square here, there is a square here. And for example, here there is a structure, there is a structure here. So we can generate large number of configurations, and the average of that will give back this kind of a contact map. Yeah.
Yeah. So from this kind of data, micro-C,
Yeah.
have people looked at like the enhancer promoter sequences and those loops, like sorry, the boxes that you see, right? I mean, this blocky structure.
Yeah.
Has it anything to do with the actual like enhancer promoter sequence? How much has it been probed?
So some of little bit has been looked at. We have some new work on enhancer promoter with this micro-C data, and Anders Hansen is the pioneer in this, who does a lot of work on this. So other than that, I don't know who has looked at it. Okay, so we can generate this contact map, and what can we predict? So we basically, so far we put in what we, and we get what we put in, but we can now predict the 3D distance between any 2 points. For example, if I take this nucleosome and this nucleosome, what is the 3-dimensional distance in nanometers? So we should be able to predict that, and we do that. And here is a prediction. So this is the genomic distance between any 2 points in kilobases, and this is the 3-dimensional distance between those 2 points after folding in nanometers. So for example, if you take two genes which are 160 kb away, and their 3-dimensional distance would be about 280 nanometer approximately. So that's what this says, and this matches with experimental data, which for example, this is an alpha globin gene. We take the contact map of that particular gene and generate the 3D distance, and it matches. If you take some other gene, it will not match, and that's what the blue shows for some other gene. Because we have to take the right contact map to get the right folding, and then only this will match. So we can predict the 3D distance. Once we have now the 3D configuration at nucleosome resolution, we can coarse grain. What does it mean? We can take, for example, 10 nucleosome and call it a big bead. So we take this 10 nucleosome and draw a spear around it, and we can ask what is the size of the spear. And if you draw many spears along the genome, we can ask what is the distance between those spears, which is LCG. And then if you have multiple spears, if you have 3 spears like this, we can draw an angle, and we can ask what is the angle between them. Now this LCG, the fluctuation in LCG will tell me what is the spring constant, and the fluctuation in theta will tell me what is the bendability. So we can, from this data which is generating large number of conformations and looking at the distributions of LCG and distributions of theta CG, in a coarse grain picture we can get some idea about the stretchability, the flexibility of the spring constant of the chromatin, as well as the bendability, the bending stiffness or bending fluctuations of chromatin locally at every point, which means along the genome we can get all this data, or we can generate this data from micro-C map. So that's what we did. So this is along the genome, the size of the spear, radius of gyration, radius of the spear, and of course it increases and decreases. Some places the radius is very high, which means that the chromatin is not folded much, it is kind of open, and some places there is a minimum, which means that location locally the genome is very highly folded. So we can get the radius of gyration along the genome for every 5 nucleosome, 10 nucleosome, 25 nucleosome, 50 nucleosome. So you tell me what scale you want. I want every 50 nucleosome, which is 10 kilobases. I have this curve of how does the radius of gyration vary along the genome. Now we can take an average of this, and that's what is plotted here, and number of nucleosomes versus average RG, and that's what this curve, and this is kind of a control, so to say, is a self-avoiding walk, which we will get n power 0.6, and real chromatin would be below that, and this is the data for real chromatin for different genes. So these are many different genes. The bluish represents the heterochromatic, and reddish represents the euchromatic regions, and there is a difference between those regions. And the dots are microscopic experimental data from Javet Zhuang's lab, and without any extra fitting parameter. The only thing is the size of the nucleosome, etc. We already know. It just falls very close to the experimental data, suggesting that the numbers that we predict are quite sensible. So this is something that we predict, again, the size of the chromosome, depending on how many nucleosomes you want to coarse grain. Now that we have it, we can do many other things, and I wouldn't go into the details of this, and yeah.
on the previous slide.
Yeah.
So this deviation that you have from the control,
Yeah.
is that basically because in the way that the chromatin is organized in the chromosome,
Yeah.
there is a return to the same points many times,
Yeah.
because the chromosome also has a sort of large structure within which the nucleosomal DNA is folded and then folded again and then brought together. So the distances can drop for both very large, the physical distances can drop for very large along genome distances, much more than for very short distances. Do you understand what I'm saying? That means that as you go away, you expect that the physical distance should largely increase with the along genome distance.
Yeah.
Right?
Yeah.
But then at some point,
Yeah.
again, there'll be a reduction because the thing will fold upon itself.
Yeah. Yeah.
Because of the compaction. So
you're right. Whenever there is a
loop behavior,
whenever there is a if you look at single cell specific structures, we will see those. These are average structures. But the reason why it is much below the green is because, as you rightly said, it is folded. So the folding alone will make it much below this. And what you said is right. When there are loops, some regions will be closer than a nearby region, and such information is already buried in this, which I am not showing now, but it will be there.
So in very large considerations, there should also be some periodicity that is observed.
But if you average over many structures, that will go away. But if you look at one specific structure, you can see some of those periodicities. Yeah. Sorry. So this is many of these things as a function of genomic location, and you can see that it has some at the TAD boundaries they have a different behavior, and interior of the TAD they have a different behavior. So this has a connection with the TAD structure. Now we can get, as you said, the distance between two nearby beads and their fluctuation, and that will give us the spring constant. So we can predict the spring constant, and so the typical number for the spring constant that you would want to put in dimensionless unit is like 1, of the order of 1 is the number I would put, or it's about 1 to 10 piconewton per micrometer. That's the kind of number that you would want to put depending on what you want to study, right? Yeah. Oh, long ago here. Yeah.
Yeah. So does this at the large number of nucleosome bits, does it go to the fractal globule?
It should show, because we have only gone up to 160 nucleosomes or so.
Right.
Because that's the only thing we could simulate. We have gone up to 400 here. We have simulated 1000. We took the central part to avoid all those end effects. So that we have, what is showing here is the scaling for 400 in the middle. So that when we average, we get all the. So there only this much one would see. But if you do like many thousand nucleosomes, one would show those kind of behavior. Yes. Yeah. So we can then get the spring constant, and these distributions, if you switch off all the interactions, analytic, we can show that it matches with the analytic relation known in when there is no interaction. Now the question is, if you have two chromatin, how do they interact? They interact, so two beads of a coarse grain chromatin, if you put an LJ, Lennard-Jones like interaction, they will not overlap. They would be like this. But in reality, these two polymer segments can mix. Therefore, the interaction between two polymer segments has to be soft, and it cannot be a Lennard-Jones interaction. So this is another point that we make, is that the interaction between any two chromatin segment in a coarse grain model has to be soft interaction, which means that segments can mix, and this we quantify now how much they can mix, and I will come to that in a minute. So before coming to the quantification of that, we can also measure the angles, just like structural biology, protein biology would do. All kinds of angle one can measure, the bending angle and the dihedral angles. So I wouldn't go into the detail, we did all that. So we have this, the bending angle distribution, and I just want to say one thing, that the distributions are quite wide, which means that the inverse width of this is of the order of kBt, therefore it is quite flexible. And then we did an inverse Boltzmann calculation to understand what is the bending potential, what is the interaction potential, and it turns out that the interaction potential is much softer than Lj. So the black is the Lennard-Jones potential, and the interaction potential between two beads of chromatin is much softer than a Lennard-Jones potential. So it has the interaction strength and the softness. So this is the idea. And then if you want to know the strength of the interaction, we can actually predict it from the contact probability. And this is very much like some of those protein ideas. And they also get something similar, that you can predict the interaction strength from the contact map by this kind of a relation. And we have, the good part is that we have this denominator slightly better fixed than many of those models. This is a parameter. And, then we can predict the Pij and the interaction strength, and then one can simulate this, and then we can get the 3D distance versus contact probability scaling relation, and it scales in a particular way, like minus 1/4 or 1/3.5, and then we can even predict the 3D distance for any two regions. So this is the 3D distance that we predict for a human chromosome, and this is the 3D distance measured from a microscopy experiments, and they are exactly the same. Note that the scale is exactly the same. It's about 100 nanometer to 700 nanometer, and the color precisely represents those 3D distances. So we can accurately predict the 3D distance for a large segments like of 2 megabases, and using this coarse grain model. I just take 2 more minutes, and just to say that 3D organization is happening one part by condensate formation, and then another part by a phenomenon called loop extrusion, where some enzymes actively extrude DNA. So they take this DNA and literally extrude loops out of it, and a competition between a loop extrusion and this condensation, or both, is basically leading to the chromatin organization. So we have a model to also incorporate the loop extrusion into the system. So the details are there in this paper, and once we do this, so we ask many questions. For example, what is the role of binding and dissociation of this protein called cohesin to obtain the 3D organization? How long would it take for this gene to organize a position in 3D, and so on and so forth? So we can ask the time question here. If you tell the rate of extrusion and the binding and dissociation of this extruders as parameters, we can know something about the time. And so one important point is that if you don't have an intrachromatin interaction, you will not get the kind of contact map that you would want. So even with extrusion, you require some amount of intrachromatin interaction. That's one of the takeaway lesson. And another takeaway lesson is that when there is extrusion and intrachromatin interaction, there can be some regions of chromatin where they compete, and then it is possible that at some locations where domain-specific intrachromatin interaction is there, this can happen in Drosophila and in some part of human, there the condensate can dominate even though there is extrusion. So effective extrusion will not be even seen, because the strong intrachromatin interaction will hold the structure, and the extrusion will have no visible effect if you look at the 3D distance, the contact map. But if you look at the dynamics, if you look at, for example, the relaxation time versus genomic separation, then the extrusion would matter, and depending on the way you extrude, you can get different results even if there is a condensate. So we claim that dynamics could distinguish extrusion versus intrachromatin interaction, while structure alone may not. Average static structure may not. So with this I will stop. To summarize, we studied the polymer property of chromatin, bead spring chain models to describe chromatin, and we predict bead size and all the parameters, soft potential, and some aspects of loop extrusion versus interchromatin interaction. I will be happy to discuss this more, and I will be happy to discuss this more after the talk and during the lunch break, and I will be happy to take questions if there is any. First row only.
Yeah, so there are models,
Yeah.
polymer models of chromosome, where they tend to take not a similar type of bit but multi-bit. That means multicolored bit.
Yes.
like 5-color bit,
Yeah.
and where each color can have the interactions which is kind of more preferred than the interbit kind of
Yeah.
interactions.
Yeah. Yeah.
So how different are your model from this, first of all? And second thing is, other thing is that it is shown that if you do not take the chip-seq data or micro-C data,
Yeah.
then what happened that you tend to see the formation of what is called knot in the polymer if you simulate. But once you put high C data or chip-seq data, then you see that those knots are not there in the chromatin structure. Have you checked the probability of knot formation in your model?
Yeah, we have checked the. I'll say second first and then later. So we have checked the knots, and we the way we fold is such a way that there's no knots. But the knot question is not at all trivial. There's a lot of things with soft interaction. In principle, there can be some knots, but even in mitotic chromosome, for example, people have seen knots. So there is recently, like Job Decker measured that in mitotic chromosome, at least the level at the scale they could measure, there are knots, and so on and so forth. So depending on, so knot question is still to be answered. It's a very interesting question that what happens and how, and presumably topoisomerase will dissolve many of those knots. So that's the knot part.
Also, high C data is very population average.
Yeah.
It is made out knots unless they occur with 100% penetrance in every chromosome.
Yeah, yeah,
They occur at the time without some.
yeah. And about the other model, of course, if you, depending on the detail that you want, so in our model, the interaction between I and J could depend on I and J, for example, epsilon ij. So if that is an effective way of saying that you have a bead, red bead, making ith bead and jth bead interact, that's a kind of an effective interaction model. And if you want some extra details of these proteins diffusing and that will do something specific, you may want to have a model like that. So depending on what question you want to answer, I think you need to take that kind of model. But if you just want to predict 3D distance, this is good enough.
Okay, so Ranjit, it's very nice talk. Last paper that you showed, of course,
Yeah.
aware of the previous stuff, but the last one where you talk about this exactly the interplay between these. So, like, do you actually consider condensation explicitly, or you're just saying that okay, the beads are kind of interacting?
Yeah, we just take intrachromatin interaction as a kind of a attractive potential.
And you were saying that underlying mechanism can be condensation or something.
Yeah.
That's the
Yeah.
because there is a paper from Frank.
Yeah.
It's on archive.
Yeah.
Where they actually look at this particular question where you have co-condensation as and looping. They
Okay.
look at the interplay.
Okay, I didn't see that.
So that might be relevant.
Okay, I'll